Hi, good morning everyone. So we're gonna discuss about the biochemical energy production. Metabolism is a total of all chemical reaction in a living organism, source of energy for the functioning of the human body, also needed for many of the cellular processes such as protein synthesis, DNA replication, RNA transcription, and membrane transport. So metabolism is one of the requirements of all living things is to help the organism or to prepare for the production of the organism. So metabolism obtains the energy and the nutrients. Subtype of metabolic reaction. Catabolism. All metabolic reaction in which large biochemical molecules are broken down to smaller ones. Usually energy is released in this reaction. Example are the oxidation of glucose. So catabolism provides energy for synthesis of the new cell function. Next is anabolism. Anabolism is an all-metabolic reaction in which small biochemical molecules are joined to form larger ones, usually require energy. Examples are synthesis of proteins, so anabolisms provide the material necessary for cell growth. So the process of catabolism and anabolism. So catabolism's reactions are exergonic, meaning they released energy. Then anabolic reactions are endergonic, meaning they need energy or they require energy, both of which are in the form of ATP. So, ang product sa catabolism can be used for anabolic reaction that is responsible for cell growth and division. Metabolic pathway is a series of consecutive biochemical reactions used to convert the starting material into an end product. So, metabolic pathway is a linked series of chemical reactions occurring within a cells that build and break down molecules for cellular processes. There are two types of metabolic pathways, the linear and the cyclic. So, linear pathway is the product of each step is the substrate for the next reaction in the pathway, while a cyclic pathway is a form of closed loop and that is the example of acetyl group. Major pathway for all forms of life are similar. So the metabolic pathway, as a knowledge of the cell structure, is essential for understanding metabolism. Prokaryotic cells, they do not have a nucleus, but instead, generally they have a single chromosomes, a piece of circular double-stranded DNA located in an area of the cell called the nucleus. Eukaryotic cells, where the DNA is found in a membrane-enclosed nucleus, about 1,000 times larger than bacterial cells. This figure shows the major organelles and other cell components of a typical animal cell or a typical eukaryotic plant cells. The plant cells has a cell wall, chloroplast, plastid, and central vacuole structure, not found in animal cells. Plant cells do not have lysosomes or centrosomes. Eukaryotic cells organelles and their function. Plasma membrane is a cellular boundary, means it is a phospholipid bilayer with proteins and cholesterol embedded in it. Cytoplasm is a water-based material of eukaryotic cells, refers to the entire region of a cell between the plasma membrane and the nuclear envelope. Mitochondria generate most of the energy needed for our cells, so mitochondria are often called the powerhouse of the cells. Next is the lysosomes. Lysosomes is a membrane-bound cell organelle. They break down excess or worn out cells parts. They may be used to destroy invading viruses and bacteria. And next is ribosomes, sites for protein synthesis. Ribosomes are made up of a large subunit, means up, and a small subunit, means bottom. During protein synthesis, ribosomes assembles amino acid into proteins. Nucleus, site where DNA is found, is the most prominent organelle in the cells. The nucleus stores chromatin, DNA plus protein, in a gel-like substance called the nucleoplasm. So mitochondrion, an organelle that is responsible for the generation of most of the energy for a cell. So mitochondrion are often called the powerhouse of a cell because they are the primary site of metabolic respiration in eukaryotic. So outer membrane is permeable to small molecules. It has its 50% lipid and 50% protein. So the inner membrane is highly impermeable. Most substances, it has 20% lipid and 80% protein, folded to increase surface area. Synthesis of ATP occurs here.
Adenosine phosphates, ATP, ADP, and AMP. Adenosine phosphates of interest. Adenosine monophosphate, or AMP, is a 1-phosphate group which has structural component of RNA. Adenosine diphosphate, or ADP, is a 2-phosphate group, key component of metabolic pathways. And adenosine triphosphate, or ATP, is a 3-phosphate groups and a key component of metabolic pathways. A phosphoryl group is derived from a phosphate ion when it becomes part of another molecule. ATP plus water produces ADP plus Pi plus H plus energy. ADP plus water produces AMP plus Pi plus hydrogen negative plus energy. And ATP plus 2 molecules of water produces AMP 2Pi plus 2H plus energy. The net energy produced in these reactions is used for cellular reactions. In cellular reactions, ATP functions as both a source of a phosphate group and a source of energy. An example of this is the conversion of glucose to glucose 6-phosphate, which is shown in the figure below. The role of other nucleotide triphosphates in metabolism. Uridine triphosphate, or UTP, is involved in carbohydrate metabolism. Guanosine triphosphate, or GTP, is involved in protein and carbohydrate metabolism. Cytidine triphosphate, or CTP, is involved in lipid metabolism. Flavin adenine dinucleotide, or FAD, is a coenzyme required in numerous metabolic redox reactions. The flavin subunit is the active form which gains hydrogen atoms when FAD is converted to FADH2. Ribitol is a reduced form of the sugar ribose. Figure 23.6 shows the structural formulas of FAD and NAD+. The next is flavin adenine dinucleotide, or FAD. FAD is the oxidized form, and FADH2 is the reduced form. In enzyme reactions, FAD goes back and forth from oxidized to reduced form. And typical cellular reaction in which FAD serves as oxidizing agent involves conversion of an alkane to an alkene. Nicotinamide adenine dinucleotide, or NAD, has coenzyme functions in metabolic redox pathways. NAD+ is the oxidized form of NAD, and NADH is reduced form. Typical cellular reaction in which NAD+ serves as the oxidizing agent is the oxidation of a secondary alcohol to give a ketone. Subunit structures of NAD. The 3 subunit structure is nicotinamide, ribose, and ADP, and the 6th subunit structure is nicotinamide, ribose, phosphate, phosphate, ribose, and adenine. Coenzyme A is a derivative of vitamin B pantothenic acid. Active form of coenzyme A is the sulfhydryl group, or SH group, in the ethane thiol subunit of the coenzyme. Acetyl-CoA is formed when acetyl group bonds to CoA-SH via a thioester bond. The coenzyme A subunit structures has a 3 subunit structure, which is nicotinamide, ribose, and ADP, and the 6th subunit structure is nicotinamide, ribose, phosphate, phosphate, ribose, and adenine. Classification of metabolic intermediate compounds. Metabolic intermediate compounds can be classified into 3 groups based on their functions. The first one is intermediate for the storage of energy and transfer of phosphate groups. This is ATP, ADP, and AMP. The second is intermediates for transfer of electrons in metabolic redox reactions. This is FAD to FADH2 and NAD to NADH. And the third one is the intermediates for the transfer of acetyl groups, which is HSCoA to acetyl-SCoA. Carboxylate ions or metabolic acids is a functional acids formed as intermediates of metabolic reactions. There are 5 such acids that serve as substrates for enzymes in metabolic reactions. These are 3 succinic acid derivatives or C4 diacid, which is fumarate, oxaloacetate, and malate, and the 2 glutaric acid derivatives, which is C5 diacid or and is alpha ketoglutarate and citrate. Figure 23.9 is the structural formulas for polyfunctional carboxylate ions. Succinic acid is a 4-carbon diacid which has a hydroxy derivative, which is malic acid and malate, a keto derivative which is oxaloacetic acid and oxaloacetate, an unsaturated derivative which is fumaric acid and fumarate. Glutaric acid is a 5-carbon diacid which has keto derivative, alpha ketoglutarate acid and alpha ketoglutarate, and a carboxyhydroxy derivative which is citric acid and citrate. Several phosphate-containing compounds found in metabolic pathways are known as high-energy compounds. High-energy compounds have greater free energy of hydrolysis than a typical compound. They contain at least one reactive bond called strain bond. Energy to break these bonds is less than a normal bond and more negative the free energy of hydrolysis, the greater the bond strain. Typically, the free energy release is greater than 6 kilocalories per mole, which is indicative of bond strain, and strain bonds are represented by the sign squiggle.
Free energies of hydrolysis of common phosphate-containing metabolic compounds. As you can see, the table has contained type, example, and free energy of hydrolysis cal per mole. The first type is enol phosphates. The example of enol phosphates is phosphenol ferrophate that have a negative 14.8 energy of hydrolysis cal per mole. And then the second one is acyl phosphates. The example of acyl phosphate is 1,3 bisphosphoglycerate acetyl phosphate and have a negative 11.3 and negative 11.8 free energy of hydrolysis cal per mole. The third one is guanidine phosphates. The example of guanidine phosphate is creatine phosphates and arginine phosphate. Has negative 10.3 and negative 9.1 free energy of hydrolysis cal per mole. And the fourth one is triphosphates. The example of triphosphates is ATP to AMP plus PP. Then ATP, ADP plus P have a negative 7.7 and negative 7.5 free energy of hydrolysis cal per mole. The fifth one is diphosphates. The example of diphosphates is PP to P to 2P and ADP to AMP plus P have a negative 7.8 and 7.5 free energy of hydrolysis cal per mole. Then the last one is sugar phosphates. The example of sugar phosphates is glucose 1 phosphate, fructose 6 phosphate, AMP2 adenosine plus P, glucose 6 phosphate, and glycerol 3 phosphates. The energy of hydrolysis of glucose is negative 5.0, and then the fructose 6 phosphate have 3.8, negative 3.8. And the AMP, adenosine plus P, have negative 3.4. And then the glucose 6-phosphate have negative 3.3. While glycerol 3-phosphate have negative 2.2. An overview of biochemical energy production. Energy needed to run the human body is obtained from food via a multi-step process involving several different catabolic pathways. There are 4 general stages in the biochemical energy production process. The stage 1 is digestion, next is the acetyl group formation, and then the third one is citric acid cycle, and the fourth one is electron transport chain and oxidative phosphorylation. Each stage also involves numerous reactions. Energy needed to run the human body is obtained from food via a multi-step process involving several different catabolic pathways. So remember, catabolic is an exergonic, meaning they release energy. So there are 4 general stages in the biochemical energy production process. Stage 1, digestion. Stage 2, acetyl group formation. Stage 3 is citric acid cycle. Stage 4 is electron transport change and oxidative phosphorylation. So each stage also involves numerous reactions. First stage is the digestion. Begins in mouth, then mag-usap siya and then mag-contain na ang saliva na siya ang mag-digest sa enzymes, then continue to our stomach, the gastric juice, and makomplete siya in our small intestine. The result of this is that in the small molecules that can cross intestinal membrane into the bloodstream. End products which are absorbed and transported to blood cells. The glucose and monosaccharides from carbohydrates, the amino acids from proteins, the fatty acids and glycerol from the fats and oil. The second stage of energy production is acetyl group formation. The small molecules from stage 1, which is digestion, are further oxidized. End products of these oxidations of acetyl certificate of analysis and reduced coenzyme NADH. This stage is naga-involve siya og numerous reaction which naga-occur siya both sa cytosol and as well as the mitochondria of the cells. The third stage of energy production is citric acid cycle. The citric acid cycle takes place inside the mitochondria. Acetyl group is oxidized to produce carbon dioxide and energy, but sometimes energy produced in this stage is lost in the form of heat. Most energy is trapped in reduced coenzymes, the NADH and FADH2. The carbon dioxide we exhale comes primarily from this stage. So diri siya nagikan ang atong gina-exhale sa diri nga stage kaning citric acid cycle. Stage of energy production, which is the fourth stage, is electron transport chain and oxidative phosphorylation. It takes place in mitochondria also. The
DH and FADH2 are oxidized to release hydrogen ions and electrons. This is needed for the production of ATP, primary energy carrier and metabolic fat waste. The of dioxygen inhaled is converted into water in this stage.
So we're now on section 23.7, an introduction to the citric acid cycle, and here are some substantial reactions that we have to take note happening during the cycle. So first, reduction of NAD and FAD to produce NADH and FADH2. So from the given statement, basically, NAD and FAD are the reduced form of NADH and FADH2. To further explain, NADH carries two electrons and bonded with a hydrogen ion. Moreover, FAD in its fully oxidized form or quinone form accepts two electrons and two protons to become FADH2. Next is the decarboxylation of citric acid to produce carbon dioxide. So flashed in your screen is the summary of the citric acid. So figure 23.11 shows a much detailed process of the citric acid cycle, which starts with oxaloacetate and acetyl coenzyme A. And here is another figure which shows the Krebs cycle, tricarboxylic acid or TCA cycle, and the citric acid cycle. So here are the reactions of citric acid cycle, which involves a total of 8 steps. So the first step is the formation of citrate, and second is the formation of isocitrate, and step 3 is the oxidation of isocitrate and formation of CO2 or carbon dioxide, which also involves oxidation, reduction or redox, as well as decarboxylation, and step 4 the oxidation of A-ketoglutarate and formation of carbon dioxide. And the fifth step involves the thioester bond cleavage in succinyl coenzyme A and phosphorylation of GDP. Step 6, the oxidation of succinate, and step 7 is the hydration of fumarate, and lastly, oxidation of L-malate to regenerate the oxaloacetate. And here are some practice exercises of 2 items. So the next slide involves the regulation of citric acid cycle, wherein the rate at which the citric acid cycle operates is controlled by the body's need for ATP, wherein when ATP supply is high, ATP inhibits citrate synthase based on the step 1 of the cycle, which is the formation of the citrate. Otherwise, when ATP supplies are low, ADP activates citrate synthase, and similarly, ADP and NADH controls the isocitrate dehydrogenase, with NADH as the inhibitor and ADP as the activator. And to make it easier to remember, the citric acid cycle also has a mnemonic, which is the citrate is Krebs' starting substrate for making oxaloacetate. And remember on the previous slide where we've discussed the 8 steps, the first step was formation of citrate, and the last step ends with the regeneration of oxaloacetate, so that explains why the mnemonic starts with citrate and ends with oxaloacetate. And to make it more detailed, just remember the products of each step. For example, on the first step, we have citric acid, isocitric acid, ketoglutaric acid, succinyl, coenzyme A, and so on. So here is another mnemonic for the citric acid cycle plus its enzymes. So, for most of the enzyme, the name of the latter consists of the molecule it modifies or the molecule directly before it and the function the molecule carries out. So, here is the mnemonic. So, at another dance, Devon slipped down. So means citrate synthetase at aconitase, another aconitase, dance isocitrate dehydrogenase, Devon alpha ketoglutarate dehydrogenase, slip succinyl coenzyme A synthetase, and down succinate dehydrogenase. So for me, it is much more confusing, but if this does work for you, then good for you. Yeah.
As the electron transport change and introduction is a series of biochemical reactions in which intermediate carriers aid the transfer of electrons and hydrogen ion from NADH and FDH2 ultimately reacts with molecular oxygen to give H2O. So the electron transfer change is a series of complexes to transfer electrons from electron donors to electrons acceptor via redox reaction and couples this electron transfer with the transfer of protons across a membrane. Enzyme and electron carriers for ETC, located along inner mitochondrial membrane, organized into four distinct protein complexes and two mobile carriers. So protein complex tightly bound to membrane. Complex 1 NADH coenzyme Q reductase. Complex 2 succinate coenzyme Q reductase. Complex 3 is coenzyme Q cytochrome C reductase. Complex 4 is cytochrome C oxidase. So there are two mobile electrons carriers, coenzyme Q and cytochrome C. Coenzyme Q, also known as ubiquinone, and cytochrome C is highly water soluble.
Complex 1 NADH coenzyme Q reductase. NADH from citric acid cycle is the source of electron processed via this complex. It contains greater than 40 subunits, including flavin, mononucleotide FNN, and several iron sulfur protein clusters. Net result is a transfer of electrons from NADH to coenzyme Q. Several intermediate carriers are involved in this electron transfer. Complex 2. It is smaller than complex 1, contains only 4 subunits including 2 FeSPs. Succinate is converted to fumarate via this complex. This complex process FADH2. COQ is the final recipient of the electrons from FADH2. Complex 3. Complex 3 contains 11 different subunits. Several FeSP proteins and cytochromes are electron carriers in this complex. Cytochrome is heme iron protein in which reversible oxidation of an iron atom occurs. Various cytochromes, site A, site B, site C, and so on, differ from each other in their protein constituents, the manner in which the heme is bonded to the protein, and the attachments to the heme ring. Complex 4. It contains 13 subunits, including 2 cytochromes. The electrons flow from site C to site A to site A3. In the final stage of electron transfer, the electrons from site A3 and hydrogen ions combine with oxygen to form water. It is estimated that 95% of the oxygen used by cells serves as the final electron acceptor for the ETC. So this is the summary of the flow electrons from complex 1 to complex 4 of the electron transport chain. Oxidative phosphorylation. Coupling of ATP synthesis with the reaction of the ETC is related to the movement of protons across the inner mitochondrial membrane. Complexes 1, 3, and 4 of ETC chain have a second function, or it serves as the proton pumps that transfer protons from the metric sides of the inner mitochondrial membrane to the intermembrane space. For every 2 electrons pass through ETC, 4 protons across the inner mitochondrial membrane through complex 1, 4 through complex 3, and 2 more through complex 4. This proton flow causes a buildup of H plus in the intermembrane space. This high concentration of protons passing through ATP synthase becomes the basis of the ATP synthesis. This figure shows or illustrates the second function for protein complexes 1, 3, and 4.
ATP formation: for each mole of NADH oxidized in the ETC, 2.5 moles of ATP are formed. For each mole of FADH oxidized in the ETC, only 1.5 moles of ATP are formed. For each mole of GTP hydrolyzed, 1 mole of ATP is formed. 10 molecules of ATP are produced for each acetyl-CoA catabolase. Reactive oxygen species, or ROS, has a greater than 90% of inhaled oxygen via respiration is consumed during oxidative phosphorylation. Remaining the superoxide is converted to several highly reactive oxygen species within the body, which include hydrogen peroxide H2O2 and superoxide ion O2, hydroxyl radical OH, and superoxide ion and hydroxyl radicals have unpaired electrons. ROS can also be formed due to external influence such as polluted air, cigarette smoke, and radiation exposure. Are both beneficial as well as problematic within the body. Example is the white blood cells produce a significant amount of superoxide free radicals via the flowing reaction to destroy the invading bacteria and viruses. So ROS is a highly reactive chemical molecules formed due to the electron acceptability of O2. ROS are formed quickly converted to non-toxic species, and about have 5% of ROS escape destruction by superoxide dismutase and catalase enzyme. So it is an antioxidant molecule present in the body to help trap ROS species, so the vitamin E, vitamin C, glutathione or GSH, beta-carotene. Major family of antioxidant phytochemical are flavonoids. So ROS are produced as a normal product of cellular metabolism. So this is the right practice exercise. Vitamin B and the common metabolic pathway is a structurally modified. Vitamin B function as coenzyme in metabolic pathways. 4. Vitamin B participate in various reactions like niacin as NAD and NADH, riboflavin as FAD, FADH2, and FMN, thiamine as TPP, pantothenic acid as CoA. So, in the absence of these vitamins B, the body would be unable to utilize carbohydrates, proteins, and fats as energy source. Next slide is the concept question.